guys, let's get started on chapter 11 on the nervous system. So, I always like to begin in having you all think about what you already know. I think we all have a good sense for the division or what the central nervous system consists of, right? So, we have the brain and the spinal cord. And then the peripheral nervous system is going to be everything else. This includes all of the spinal nerves as well as the cranial nerves. When we first looked at the systems, I drew a contrast between the nervous system and the endocrine system. In both cases, they are systems of the body that have the ability to communicate across the body. And with the nervous, we see that it is fast acting and very specific. Whereas with the endocrine system, those hormones, because they're traveling through the bloodstream, they're going to be a little bit slower to act. So, where are we going with this? So first of all, just to give you an overview, I want to talk about the organization of the nervous system. How is it structured? What are the different divisions? And then we'll move on to histology. So the main players here are going to be neuroglia as well as neurons. And so I'll look at the neuroglia in the central nervous system first, and then we'll cover the ones in the peripheral nervous system. Then we focus in on the movers and the shakers of the nervous system, if you will. These are going to be the cells, much like muscle cells, that are electrically excitable, right? So they're going to be able to generate an action potential, which I'll save for the next video. We'll look at the structure and function of them, and we'll talk a little bit more about myelin. And in doing so, I want to draw a contrast between multiple sclerosis, which is a progressive disease that we've all heard of, and I'm sure many of you have a great deal of familiarity with, but it is progressive, it's autoimmune, it tends to lead to muscle weakness, tremors at times, but it can also affect the vision, memory in some cases. In contrast, Guillain-Barré is a condition that has a very quick onset. I've actually had two good friends who have experienced this or have suffered from Guillain-Barré, and it progresses as fast as going to bed healthy and waking up without being able to move your limbs. So I want to look at some of the similarities and differences between those two very different conditions. And then last up, just a slide that looks at some key vocabulary. So gray matter versus white matter, what's a tract, what's a nerve, and how is that different from a neuron? And then last, ganglia versus nuclei. So here we go. First up, looking at the organization of the nervous system. So again, central nervous system, peripheral nervous system. So in this picture, to orient you, here's the central nervous system, right? So this is going to be responsible for integrating sensory input, for interpreting it, and implementing action, right? So the action that it sends out is going to go via motor pathways. And those motor pathways are also referred to as efferent pathways. The sensory information, sensory input coming from all of our senses, from internal as well as external input. In those cases, sensory is going to be the same as afferent input. The only difference between these two words is an A versus an E. So I always think A for approaching the central nervous system and E for exiting the central nervous system. And that can be a nice way to keep those two straight. So with the central nervous system, to elaborate a bit, right? So you've got, this is going to again be the integration and control center. It's interpreting that sensory input and it's dictating that motor output. On the peripheral nervous system side, so this includes those afferent as well as the efferent pathways, right? So again, you've got your cranial as well as your spinal nerves that are going to be involved here. So I've got a couple different graphics that show you how we can organize this. So in this one, central nervous system is shown here at top, and I think that makes sense, right? It's kind of the higher order processing. And then the peripheral pathways are shown down here in this yellow box. So sensory information through the afferent, right, approaching division. So much like what we saw on the last slide. And then motor commands are going to travel through those efferent pathways. So let's build on this a little bit more. So from where does that sensory information come, right? I alluded to this idea that it's taking in both internal as well as external information. So just pause and think to yourself for a second where that may be coming from. And I suspect that many of you are thinking, okay, five senses, right? So eyes, ears, tastes, etc. But what we other systems that we're going to consider here are going to be your somatic sensory input. So proprioception, information about that's going to communicate information about our joints, about our body position, and then also visceral sensory receptors. So your gut, right? So if you have stomach upset, if you have period cramps, right? Those are going to be communicated through those visceral sensory pathways as well. So coming down here on the efferent side, I'm layering some more details here. So you've got the somatic nervous system. So soma means body. And then you have what we call, the autonomic nervous system. So autonomic, you want to think automatic, right? So if we don't have any control over the system, we can consider it to be involuntary. In the case of somatic nervous system, this is going to be voluntary. So think about the tissue types that we've covered and what sorts of efferent effectors are we going to see pop up in this box. So see if you can kind of guess what those would be. Right, so with the somatic nervous system, since it's voluntary, we're acting upon skeletal muscle. And then over here with our autonomic, right, so automatic, you've got smooth muscle, cardiac muscle, and you also have glands. So all of your mammillary glands, sweat glands, right, salivary glands, those are all going to be controlled via involuntary pathways. We have a whole chapter looking at the autonomic nervous system. I'm sure these divisions are very familiar, right? So sympathetic is going to be fight or flight. Parasympathetic is going to be rest and digest. It's kind of a kitschy way to remember those two. So yet another graphic that looks at this division. So I think this is obviously something important, spending this much time on it. So really getting this organization in your mind, I think, is going to help. Simply because we do have separate chapters looking at the autonomic nervous system and looking at its various divisions. So with this graphic, notice central nervous system is over here, right? And we're really focusing in on the peripheral nervous system. Here is the sensory division, right? So approaching, and then the motor division, right, is going to break up. Just like we saw in the last slide, you've got somatic and you've got autonomic. Here are those pieces about voluntary and involuntary. And it just gives you some more details. I like that in this graphic, you also have some examples, right? So somatic sensory, I mentioned proprioception coming from our joints. Certainly touch, hearing, sight, all of that is going to be through this pathway as well. Visceral, right? So you're just getting a few more images here and a different way of presenting this content. So whichever graphic sits best with you, but I think they're both valuable ways of appreciating how our body's nervous system is organized. Okay, so moving on to histology, right? So you've got two main cell types. Like I said, the neurons, these are the excitable cells that can pass electricity, if you will. And then in addition, you have what are called neuroglia, sometimes just glia. And glia means glue. So we have really gained an appreciation for how critical these neuroglia are in supporting our nervous system over the years. They used to be dismissed to some extent with all the focus being on the neurons. And then what you have here is actually a nerve. And I really worked hard to contrast the difference between a neuron and a nerve. So this is a nice histological image of a nerve. And what you're actually seeing here is a cross section. So any one of these, if you were to take our neuron here and make a slice right across here, right? You've got the cell itself, the axon, and then you have the myelin that surrounds it. And so this dot in the middle is that axon, The white around that, if you can see where I'm pointing, is going to be the myelin. And then the pink border, if you will, and we'll define all these terms, is going to be this connective tissue that surrounds it. So we're going to see a lot of parallels here with the organization of the nerve and the muscle in terms of all of the connective tissue that wraps around it. So neuron versus neuroglia. So I'm going to start off elaborating more on these neuroglia, and in doing so, focusing on the central nervous system first. So astrocytes, these are the ones that are most prevalent in the central nervous system. Astro meaning star, right? So look at this star. Look at this star shape of this cell, right? Extensive branching. And if you look at what it's doing, these are such slick cells. So they have extensions, podocytes, that are wrapping around the capillary, and then they have other extensions that are wrapping around the neuron itself. So they are actually serving to pass food and nutrients from your bloodstream to these neurons. So think about that, right? In contrast to other tissues that we've seen, the blood does not directly feed neurons in your central nervous system. And so, in essence, these astrocytes are forming part of what you've heard of as the blood brain barrier. And they're just a piece of it, but they're a really important piece of it. So, those are going to be the astrocytes. Microglial cells. So, as the note indicates here, these are part of your immune system. So, they're a special type of macrophage that help to clear bacteria or other invaders that may enter into this space in your central nervous system. In addition, we also have ependymal cells. So, if you look, this is being labeled as the brain or spinal cord tissue. And this is a fluid-filled cavity. And so the fluid that's filling this cavity is your cerebral spinal fluid. So we'll look more at this in the central nervous system chapter. But there is these ependymal cells basically create this sort of like a barrier between your cerebral spinal fluid and your brain. So in bacterial meningitis, right, these ependymal cells are actually compromised. That's one of the reasons that it can progress so quickly and dangerously. And then last up, oligodendrocytes. So these are cells in your central nervous system that lay down the myelin. So we're going to see a different type of neuroglial cell laying down myelin in the peripheral nervous system. So those are they're the rest of the terms, oligodendrocytes, right? So those are the 4 glial cells that you'll need to know for the central nervous system. There is also a video link here that you can play to reinforce much of what I was just saying. And then with the peripheral nervous system, there's 2 that you need to know. So you have satellite cells and Schwann cells. So satellite cells are shown here, wrapping around the cell body of this neuron. And then in this case, the Schwann cells are those that are forming the myelin around the axon of the nerve. Cool graphic from your textbook. I like that it's showing how this Schwann cell is able to wrap around the axon. Here's the nucleus here, and it begins to wrap, and some of the cytoplasm is basically forced from between the membrane, as the textbook describes, and you get this really tight wrapping going around the axon. So just a cool graphic, I think, again, to illustrate what these myelin sheaths are really made of and how it is that they form. With the neurons. Okay, so specialized cells that conduct impulses. So muscle and neurons, right? Those are really muscle cells and neurons. Those are really the only cells that we see in the body that have the ability to essentially change their membrane potential. Other cells in our body, that membrane potential, the charge across the membrane, stays constant. But with these cells, they have the ability to transmit action potentials. And in doing so, they're essentially communicating, right? So you're going to see just basic organization here. So here's the cell body, nucleus. Soma means body. And then along the axon, notice that you don't have myelin wrapping around this portion of the cell. This is going to be what we consider to be the receiving end of the cell. It's going to pick up on signals. And the role of myelin is really to insulate and to help transmit impulses. So it would be counterintuitive to have myelin wrapping around these dendrites when you're trying to enhance communication at that point. And then last up are these axon terminals. If you think about the neuromuscular junction, right, at the end of that neuron, we saw, I think about our synapse and our presynaptic terminal. That's the exact same thing that you're seeing here. So at the end of these terminals, you are, of course, going to have neurotransmitters of various kinds, right? Here's this difference between a neuron and a nerve. So I wish I could zoom in on this a little bit more, but here is a cell, right? This is the axon. This is the myelin sheath around it. And this whole thing is the nerve, right? So the nerve includes blood vessels. It includes connective tissue. It includes all of that. Whereas the neuron is a single cell, typically in this format, right? With a cell body and an axon. In your study guide for lab, you'll see these terms endoneurium, epineurium, and then let's see, perineurium. There it is. And endo, peri, and epi, right? Those parallel directly the terms that we saw for the muscle, right? So wrapping around the individual muscle cell was that endomysium. In this case, wrapping around the individual neuron is going to be the endoneurium. And then we see this organization where axons do bundle in groups or fascicles. So a lot of repetition here. And those fascicles, this group, is surrounded by the so periscope going around neurium. And then last up, these fascicles come together in groups to make up this entire nerve. And that nerve is surrounded by the epi, meaning surface neurium. So in the case of the muscle, right, the counterpart there was the epimysium wrapping around that whole muscle and oftentimes being continuous with a tendon. So I wanted to point out that organization while I had this graphic up. And this is taken from your book, so you can see a blown up picture and visualize it a bit more clearly. So some things to consider about the physiology of neurons. So they have a great deal of longevity, right? If you contrast that with epithelial cells, right, which epithelial cells in our tongue or our mouth or in our gut or our skin are replaced constantly, right? A high degree of turnover. They're not intended to have a longevity like this, so 100 plus years. So that's the good news, but the bad news is, right, they don't divide. They're not designed to rapidly divide. And the few exceptions here, interestingly enough, are your olfactory bulbs, so your sense of smell, and hippocampus, so memory. And there's some cool connections there in the central nervous system that we can talk about as well. But for other than that, right, our neurons really don't divide. They don't repair. They don't have the ability to regenerate. And so this is where you really can think about the implications for spinal cord injury, right? How it is that an injury can be so permanent. Neurons have a high metabolic rate. So even though they don't divide, that doesn't mean that they're not continuously using oxygen and glucose. So they are fairly greedy in that regard, similar to the muscle, right? They're going to use a lot of energy in order to function. Just a different way of looking at the path of information flow through these neurons. So again, your cell body, your axon. Here are your axon terminals with the neurotransmitters at the base. So you can think about this end as being the receptive or receiving end. So I love this graphic because of the way the arrows are shown. So information is coming in through these dendrites, collecting electrical signals. It's integrated through the cell body. We'll talk about the relevance of a structure here called the axon hillock. And then assuming you have, assuming you reach threshold, you'll have an action potential that travels down this axon that's going to pass those signals to the dendrites of another cell, or another effector cell, right? So bear in mind that this could be muscle, it could be a gland, it could be another neuron. So those are the key examples there. And so this is going to be the conducting region. One thing that I think is helpful to think about here in talking about the skeletal system and in this system, those are two systems where I think it can be sometimes easy to think about them as being relatively stagnant, but please don't forget how adaptive the body is. All systems are incredibly adaptive. And so in the body of these very specialized cells, right? We have endoplasmic reticulum, we have all these different organelles. And in the case of rough ER, which makes proteins, right, it is very active. So the nucleus of these neurons is carrying DNA that's going to code information that essentially is going to make things like neurotransmitters, that's going to code for sprouting different receptors. Those are places in the neuron that can be adaptive. So even though the neuron, the cell itself is not going to regenerate if it's damaged, that doesn't mean it's not still adapting to the environment. So it can still change its functionality, even though it's going to be consistent in terms of its own longevity. With the functional classifications of these neurons, so very similar to what we saw before, I'm even using the same graphic, but here I'm only classifying the cells themselves. So you've got neurons that are dedicated to the sensory input, right? So somatic sensory cells carrying information from the gut and the visceral, from the body, and then with the visceral receptors carrying information about the gut and the GI tract. And then again, the neurons in the motor system, so somatic, voluntary, and then the visceral is going to be involuntary. So this would be something like controlling peristalsis in your GI tract to move food through your large intestine or small intestine. And I think it's interesting to consider whether or not we have more neurons dedicated to sensory perception or motor control. So just pause and guess for a second. Try not to look ahead, right? And I think a lot of students are surprised by this. Look at that. Right? Of course, these are average numbers, but about 10 million of our neurons are going to be dedicated to sensory input, as opposed to about 500,000. And if you think about all the divisions on the motor side, right, we had the autonomic, we had sympathetic and parasympathetic, so it seems like a lot. But if you consider how involved sensory input is, how much goes into visualizing our world, into hearing our world, into navigating our vestibular system and interpreting that, right, coming from our ears, it's remarkable. And so we have far more neurons dedicated to this side of things. And then lastly, the interneurons I haven't alluded to yet, but we're going to see that they reside in the central nervous system. So they'll come back into play when we look at reflexes. And they, as it implies here, they're going to help integrate the sensory input with the motor output. So that because they play that role in integration, it's fitting that they're going to be in the central nervous system. So last up, I want to look at myelination. So what is myelin? Right? We've looked at the cells that make up myelin in both the central as well as the peripheral nervous systems. But what it is white. So it's a protein lipoid substance. So protein fat substance. And it's going to be white in color. And I don't know, I in my head, I'm thinking a lot about development and the final month of pregnancy. Most of the brain is made up of fat, and so There's a great deal of laying down myelin that takes place in those end stages of development, and a lot of fat is needed for that. Similarly, just as the brain continues to develop, you're going to see how important fat is, in large part because it helps to develop the myelin in the brain. So, in a myelinated fiber, I like to think a nice analogy here is thinking about electrical tape wrapping around a wire. So with that electrical tape, it helps to insulate that the passage of electricity, and it's going to make the passage of electricity more efficient. And it's very much the same way here. So the myelin is going to allow for that communication to occur rapidly and efficiently. So there are fibers in the body that are not myelinated, and they're going to conduct impulses much more slowly. And so this brings us back to this clinical application that I wanted to make between multiple sclerosis and Guillain-Barré. So with multiple sclerosis, what's going on is it's the myelin in the oligodendrocytes, those that make up the central nervous system, that is targeted. So it's basically a central nervous system with the MS versus a peripheral nervous system with the Guillain-Barré. So even though Guillain-Barré can progress very, very quickly, right? This was the example that I gave of going to bed okay and waking up with some degree of paralysis or quite a bit of paralysis, right? These individuals can make a full recovery, and that's largely because it is in the peripheral nervous system and it's not progressive like it is with MS. With MS, it's central nervous system, and so that's why you see some of the influence on things like vision, memory in some individuals. Yeah. Okay. Last slide for you is looking at organization once again. So, central nervous system, peripheral nervous system. So, gray matter versus white matter. So, as I'd indicated, one way to think about this is white matter, right? It's white because of the myelin. And so if you look at we'll start with the peripheral nervous system here. So consider the structure of an individual cell, right? You've got the cell body and then you have the axons. Well, that's a single neuron, right? And so as those neurons come together in the peripheral nervous system, those axons, so think about balloon strings. Sorry, an analogy is coming back to me. Think about a balloon as a single neuron. Now you grab a whole bunch of balloons, and the balloons themselves will be like the cell body, and the strings are going to be like the axon. You guys with me? Okay, so if you think about the bundle of cell bodies in the peripheral nervous system, we're going to call this collection of cell bodies ganglia. And then in contrast, the strings of the balloon, if you will, those are going to be the axons. When those come together, that is what we call a nerve. Okay? So, and because it's with those axons, right, because they're typically going to be lined with myelin, that's why it's broken up here as white matter. And the cell bodies themselves don't have myelin, right? We want to enhance the communication, so gray matter there. So hopefully that makes sense. And then coming over here with the central nervous system, so a lot of different terms here. The ones that I have listed here are the ones that I'd like you to know. Okay, so with the central nervous system, let's go back to our balloon analogy. So again, the balloon being the cell body. So when you have this collection of cell bodies, and those cell bodies are located within the brain or spinal cord, we call those nuclei. So it's ganglia in the peripheral nervous system and nuclei in the central nervous system. Meanwhile, the axons, when they come together in the central nervous system, I like this description, they share a common origin, destination, and function, right? So think about your optic tracts. Those are called tracks. right? Those are going to be the bundles of the balloon string. And then, yes, I think that's a good stopping point. So hopefully that was helpful to you all. I do have some review questions. In the next video, I'm going to be jumping into communication and graded potentials versus action potentials. So let me put these questions up on the board, if you will, here for a second. Maybe you just want to pause the video here and take a stab at answering these questions. So I'll go ahead and answer for you. So while studying for an exam, you reach for a beverage. To extend your arm, your blank nervous system is activated. So it's voluntary. So skeletal muscles get being activated. And so it's going to be somatic. Coming down here, complete the analogy. Electrical wire is to electrical insulating tape as peripheral neurons are to, so which cells are surrounding, making the myelin? So it's going to be the Schwann cells in the peripheral nervous system. And then this one here, door slams shut loudly, you flinch. After a few seconds, you realize your heart is beating rapidly, forcefully. So you may immediately be thinking fight or flight or your sympathetic nervous system. And that's great, that is correct. And you just want to generalize that and recall that that sympathetic nervous system is categorized as one of the autonomic branches of the nervous system. So I will stop it there. Thank you guys for your attention. Hopefully that was useful. Just let me know if you have any questions.